So we have this system set up. We have the pump, we have the tubes, we have the fluid. But how does stuff actually get from the tubes out into the tissue spaces? This is what happens at the capillary. So this is capillary exchange. And let's look at how this works. So for capillary exchange, let's say we have our blood flowing here, and it's coming from our artery. And then it has to proceed into an arteriole, which controls the distribution of blood flow. And then from this arteriole, it goes into the capillaries. So if we drew out a capillary here, and let's go ahead and draw it as a tube. So if you imagine this is the tube of the capillary. And it has two ends to it. There is one end that is close to the artery side, and we just call this the arterial end. And then there's one end that leads into the veins, so we call this the venous end. And then the venous end would lead into, as we talked about, venules, and then these would be collected into veins, which would then take the blood all the way back to the heart. So what forces are at play here? I need to get substances out of the blood vessel so that they can actually go to these cells, these tissue cells that need all of these substances. And this capillary system is incredibly important. There is no cell in the body that is more than approximately 100 microns away from a capillary. And this idea of a micron is a little hard to wrap your mind around. But if you think about and you look at the hair, either from your head or say from your arm, a typical hair is about 100 microns in diameter. So this is a very, very short distance. The heart is pumping blood. And what it gives to that blood is a pressure. So we have blood pressure. And we talk about blood pressure in terms of a hydrostatic pressure. And hydro simply means water, and static is not moving. So we measure these pressures assuming that there's no movement of fluid. So we call it hydrostatic pressure. So this is what blood pressure is, this physical force that is trying to push stuff out of a vessel, out of a capillary. So as blood comes through here on the arterial end, we can imagine if we draw in an arrow, for instance, that this arrow is representing this hydrostatic pressure of the blood pressure, and it's pointing towards the vessel wall. So in reality, right, I'm only drawing one arrow here, but this blood pressure is pressing on all of the walls of the capillary. So it's this three-dimensional pressure. But this is the major force that is trying to move fluid rather, out of the capillary. So this is our, I'll abbreviate it, P, pressure, hydrostatic. Now what's the other major pressure that's at play here? This is a pressure that's more difficult to conceptualize, but there are proteins. Remember I mentioned that plasma contains these proteins, and the proteins are within the blood, and they are large proteins. They are not able to easily leave the capillary. So these proteins, the most abundant one is called albumin. So this, the fact that the proteins can't leave the vessel, combined with this idea of osmosis, leads us to our second pressure here, the second major pressure we have to consider. Osmotic pressure is a pressure that is exerted that tends to draw fluid towards where there are solutes, where there are proteins. This is the idea that water follows salt. Well, you can also say that water follows large proteins. So the large proteins here are present within the capillary, but they're not in high concentration outside the capillary. What this means is that overall, there is an osmotic pressure that tends to try to hold on to fluid in the capillary. So I'll abbreviate this as pressure osmotic. Now, on the arterial end of the capillary, the hydrostatic pressure tends to be higher than the osmotic pressure. So overall, if you think of this like a tug of war, the hydrostatic pressure is winning, and it's trying to push fluid out of the capillary. And it does so. So here we would have water that's coming out. And along with that water, it takes all its goodies too. So it has nutrients like glucose, for example, or the ions that are in blood. And it's delivering these substances to the tissue cells that are residing out here. So I drew them on the other side, but I'll draw one right here. Okay, this is great. But this presents a potential problem, because if you drain the blood of its fluid component, then one, it gets too thick, but also the whole volume plummets. So you have to have some way to recover this water that has delivered the nutrients out to the tissue cells. And we can do this with a different balance of pressure on the other side of the capillary. Now, for the most part, these plasma proteins, this albumin, doesn't escape the capillary. So the size of its pressure stays the same between the arterial side and the venous side. So this osmotic pressure does not change. So what does change is this hydrostatic pressure. And if you think about the fluid moving through a hose, for example, the fluid at the very start of the hose has a higher hydrostatic pressure than when it gets to the end of that hose. And as it's moving through this tube, it loses its energy, it loses its pressure, because it has to rub against, it has to flow through and contact the walls of that tube. And that creates a surface where energy is lost. So pressure is lost over a particular distance. In the same case, right, the vessels are like tubes, are like a hose. And here, by the time you get to the venous end, this hydrostatic pressure has gotten smaller. And at the venous end, the hydrostatic pressure is actually lower than the osmotic pressure. So now for this tug of war, the osmotic pressure is winning, and it tends to draw fluid back into the capillary. So we can have a conservation of this fluid volume in the system. Now, it's not 100% efficient, though, right? So overall, we have approximately, and this figure varies from person to person, but we'll say that on average, over the course of a day, there's between anywhere from 2 to 8 liters of fluid that is not completely reabsorbed into the venous end of the capillaries. So you can imagine that this is a tremendous volume. And how is it handled? Well, we'll talk about that next. So the system that handles this excess fluid that is not taken back up into the blood capillaries on the venous side is the lymphatic system. Now, the lymphatic system is a completely distinct set of vessels within the body. And their purpose, one of their many purposes, is to pick up excess fluid that is in the interstitial space, in this space between the tissue cells. And they have a very cool way that they work. So they have a system of cells that are overlapped, and they serve as flaps that are anchored to the tissues that are surrounding them. So there are these anchoring filaments right here. And these anchoring filaments sit out in the bulk of the tissue. So they're situated in a way that allows them to be sensitive to any sort of volume expansion. If the tissue expands in volume, then it pulls on these anchoring filaments. These anchoring filaments are attached to those flaps, so now they pull the flaps open, and when they do that, fluid can then go into the lymphatic vessel, into these specialized lymphatic capillaries. Now, don't be confused by this terminology of capillaries, right? These are completely separate vessels from the blood capillaries. In this diagram right here, it's showing on the left the arterial side, and then here in the red and blue are the blood capillaries, and then here is the venous side, the venous side of the circulation. These green vessels are a completely separate set of vessels that interweave between and alongside of the blood vessels. And they are largely responsible for taking in this excess fluid that leaks out. So then what happens? Well, you have these flaps that open, and the fluid can go in quite easily. But another really cool thing about the lymphatic system is it serves as a highway for larger materials like cells and particularly white blood cells and some bacteria too, that can then come into the lymphatic vessels, where they are then sent back to the venous circulation. So these would lead all the way eventually, ultimately, back to the vena cava, so that then this fluid can be conserved into the blood. So how's this system set up? Well, remember we had a systemic side of the circulation, and then we also had a pulmonary side to the circulation. And the lymphatic system sits alongside these circulations. So the fluid can be picked up into these lymphatic capillaries right here. So here are the lymph capillaries. And they are taken to special structures called lymph nodes. These lymph nodes have a high concentration of white blood cells within them. So there are immune cells there, immune cells, that can now screen whatever substances the lymphatic system is carrying. So any sorts of bacterial byproducts, toxins, the bacteria themselves, viral particles, parasites, they get transported through the lymphatic vessels to these lymph nodes, where your body can mount a response in case it needs one. Now, if everything's A-OK, then the fluid can then return to the venous system, ultimately, where there are distinct points where the lymphatic vessels drain right into a venous vessel. So there are points where the lymphatic system has to directly connect to the blood vessel system in order to allow this fluid and any sorts of larger particles like proteins and cells to re-enter the blood. And this happens so that the blood can then maintain volume and get circulated back throughout the entire body.